Okay, so then, Luca, the floor is yours. Thank you very much, Emilio. Thank you all for being here today to listen to this session, which will be devoted to an innovative approach for the biocontrol of fermentation processes, thus exploring the potential of microorganisms in view of controlling undesirable yeast during fermentation processes, and especially during the very early stages of the fermentation processes. You know very well that there's a fight currently going on chemical preservatives, and the entire industry is striving to find solutions to reduce the use of preservatives to then not have to indicate their presence on the labels of foods. That is a process that exists in the world of wine, because you know that there are harsh battles being fought on SO2, and as you shall see by the end of my presentation, there are alternatives that may not be as ideal as they sound, because I think we can find that we can strike the right balance between the use of microbial activities and the use of chemical preservatives. Now, before we deal with the presentation and before I address the experiments we've carried out with our colleagues of CHR Hansen using their strains, I like to highlight that the world of lactobacilli, the world of lactic acid bacteria, has undergone a far-reaching revolution in recent years. Especially, there's one colleague of ours at the University of Verona who participated very actively in a recategorization of the taxonomy. Professor Fellis of the University of Verona contributed very actively in what was the reordering of the Lactobacillus genus, which was reclassified after in-depth genetic examinations. It was reclassified into 23 novel genera, one of which is of interest to the wine world, because the Lactobacillus plantarum, as we were used to calling it, has changed its name. It's called Lactiplantibacillus plantarum. So the terminology needs to be updated. This has no impact on its technological use, because the microorganism is still the same and its application and potential are still the same. But from a taxonomy perspective, it needs to be called differently. So then, talking to Giovanna, we were saying that one of the suggestions from the scientific world to the industry was not to change the initial of the genus. So L. plantarum will remain. In the past it was identifying a Lactobacillus plantarum. Now it's Lactiplantobacillus plantarum. So lactic acid bacteria are very heterogeneous, and many of them have a role in wine. The most representative are Enococcus, Lactobacilli, and Pediococcus. If you look at at their role during fermentation, it is clear that their role of biodiversity is very important in the early stages of the fermentation, while following fermentation pressures, because fermentation is a process where interactions are created among the various microorganisms, and especially the strongest take over, then things change. When a Lactobacillus cerevisiae or an Enococcus takes over, it means it has a role to take to dominate the fermentation process. When fermentation processes go on and the competitive pressure and the stressogenic pressure continues as provoked by alcohol, there's one population that becomes prominent, and that is Enococcus ani. There are many microorganisms that luckily enough are inhibited. When we talk about Pediococcus, we're talking about Hilgardi or brevis. We know that heterofermentation in wine is not working well, because then it will lead to what is typically known as wine diseases. But Enococcus ani is the most capable, the most talented that can resist the fermentation conditions. So it's the main malolactic microorganism in the traditional meaning of malolactic. If what you see is a classic trend of a malolactic process, where first you have an alcoholic fermentation and then a malolactic fermentation happens, you see that when the competitive pressure of the yeast is lower, there's a growth in the population of lactic acid bacteria, because there's a loss of competitive pressure for Saccharomyces cerevisiae, leaving enough room for other microorganisms to develop. And so the lactic acid it will increase, malic acid will go down, and the rest is on the slide. Now, these traditional conditions have been accompanied by the so-called co-inoculation of lactic acid bacteria with the yeast to drive fermentation processes that at the same time will consume sugar, which is consumed by Saccharomyces or yeast, and a malic acid consumption by lactic acid bacteria. This has created several questions associated with the ability of the yeast to control the attack by lactic acid bacteria on sugars, because Enococcus ani is a typical heterofermentator, so it can produce acetic acid under certain conditions. But if we implant incorrectly a strong strain of yeast, then that is able to rule out the contact by the lactic acid bacteria on the sugars that are redirected, and in the matter of 7 to 10 days, the process will come to an end. And after that period of time, the wine can be stabilized and later bottled. On top of this new use of lactic acid bacteria as a co-inoculation for fermentation processes, we're exploring the concept of bioprotection. So the use of microorganisms and is mostly focusing on lactic acid bacteria, but not exclusively those, to control the indigenous populations that will come from the field and from the grapes, which we know is well connected to processes of quality deviation in fermentation processes, because we know very well that you have wild yeasts such as CHR Hansen, which produce a lot of acetic acid, and we have acetic bacteria that will come directly from the field, especially if the grapes are not under perfect health conditions. So the tool that winemakers have at their disposal at the beginning of the fermentation process is the addition of SO2. But in line with the philosophy that points to a reduction in chemical preservatives, we thought of how to exploit competitive abilities on the part of wild microorganisms to inhibit the growth of these populations, which are unwanted. The area of bioprotection originally came in the animal product, animal foods for against pathogens. So lactic fermentation, which is very common in dairy products and fermented meats, cured meats, was seen as a very interesting tool that could be used in order to reduce the concentrations and eliminate technically the pathogen concentrations in these animal foods, animal-derived foods. In wine, luckily enough, we don't have any pathogen issues, because if you inoculate Listeria or Salmonella in wine in a matter of a few hours, they will probably be eliminated, probably even after just a few minutes, but we may have problems in terms of the development of unwanted microorganisms. And here comes the concept of biopreservation. This is perhaps the first scientific paper where biopreservation or bioprotection has been described by this researcher, a Canadian researcher, in connection mostly with lactic acid bacteria, but the idea has expanded because any microorganism that can fight unwanted microorganism can be regarded as a bioprotective agent. Well, we talk about metabolites and even the simple presence, because microbial environments need to be colonized, and so the competition in terms of the colonization of this environment is another area that can be developed. In this area, strategies differ widely. We can study bioprotection under laboratory conditions that highlight the various relationships between the various populations of microorganisms in order to understand the mechanisms that underlie bioprotection. In the world of winemaking, we can go as far as the vineyard, and there are some applications that concern the use of certain yeasts, especially Meshnikovia, that produce killer toxins for botrytis, against botrytis cinerea, and widely used also in the fruit production sector, that has opportunities to offer in terms of protection of grapes, to then go to the fermentation processes, where, as you can see, we can use both yeast and bacteria in order to control unwanted populations of yeast and bacteria. Among these, the microorganism that we have worked with in collaboration with CHR Hansen is Viniflora nova. Viniflora nova was proposed as a malolactic agent, but because of its bioprotection characteristics, it was also proposed as an agent to inhibit bacteria and yeast, and therefore, from this point of view, it can initially control the unwanted populations and then possibly also give in a possibility to control malolactic fermentation. From this point of view, we followed an experimentation both in the harvest year 2019 and harvest year 2020. We worked at the laboratory level. The data I will present come from micro-fermentations on small volumes, and then Duncan will present data where we have sort of industrialized the possible applications. As you can tell, in 2019 we worked with a control fermentation, and then later on we added 5 grams per hectoliter of SO2, and then a third sample where we only inoculated Lactobacillus plantarum without SO2, to then expand a little the experimentation in 2020, when we tested several doses of sulfur dioxide and the combination between sulfur dioxide and the use of the bioprotective agent. We recreated in the lab the microbial ecology of a must, adding Gansiniaspora, Citobacter, Stamarella, Meshnikovia And then we also obviously added Saccharomyces cerevisiae as a fermentation agent. Wherever we had added sulfur dioxide or our Lactobacillus plantarum. The composition of the master was identical for both vintages, and in 2020 we recreated the same conditions as we had found, as we had encountered in 2019, just to take a homogeneous approach. And we followed the fermentation in terms of microbial count as well as the composition of the wines. These are the 2019 results. There certainly is an impact from sulfur dioxide. Do you know if I have a pointer? Okay. So you see there's an impact on the part of sulfur dioxide in the population of Acetobacter and Hansen which was expected, because essentially they decline much more quickly in the presence of sulfur dioxide. When you only have lactobacillus, you see that lactobacillus will tend to reduce the concentration of both populations, although not so effectively as in the case of sulfur dioxide. A more important impact on the population of Meshnikova, where you see that with sulfur dioxide it goes down more quickly, and Meshnikova is known to be not very resistant to SO2, and the other bacterium Stamarella resists to these concentrations. So when I inoculated the lactobacillus manages to coexist with Saccharomyces for at least 5 to 6 days. What is pretty interesting in this experimentation is that you see the density that Saccharomyces cerevisiae can reach, which is maximum when you add sulfur dioxide. So the addition of 50 milligrams per liter of sulfur dioxide can inhibit the competitive populations, leaving more room to Saccharomyces cerevisiae, which grows more. So in both the sample without sulfur dioxide and in the one with Saccharomyces, with Lactobacillus plantarum, you see that there's a different result. Here you cannot see the results, I will tell you, because there's a black box covering the slide. Essentially, what we wanted to show in this slide was the control of both the population of Acetobacter and the population of Hanseníaspora. So behind this box, there were very nice bar charts, all colored, where we would have shown that in the presence of SO2, Acetobacter and Hanseníaspora are the populations that are immediately eliminated, but also the Lactobacillus plantarum can control them. So there is a significant difference between the loss of Acetobacter and Hanseníaspora populations versus the control. And that is very clearly visible here. When we measured acetic acid and ethyl acetate, whenever we had a control population and a spontaneous fermentation, you see that the figures were very high. When we inoculated Lactobacillus plantarum, the figures were very, very low, and especially for ethyl acetate, there are no significant differences versus the samples where we used the sulfur dioxide. From a sensory perspective, more generally, considering the volatilome, and these results were obtained from a gas mass analysis. The three samples were totally different. With respect to certain indicators written here, like linalool, glycerol, 1-propanol, that could differentiate the different samples. Then we repeated that in 2020. In this other case, we added several concentrations of sulfur dioxide. Once again, there's a very clear impact on the population of Acetobacter and Hansen which was totally expected. Once again, an impact on the decline in Mechnikovia populations. And once again, you can see that with 3 grams per hectoliter and 5 grams per hectoliter, the Saccharomyces populations managed to grow more than the wild samples. When you add the lactobacillus, well, this was essentially the same as what was mustered before. So what's interesting here is that 6 days later, we have a total elimination of the unwanted populations, and the decline is associated with the concentration of the sulfur dioxide. 5 grams per hectoliter is certainly the one that shows a faster decline in Acetobacter populations and Hanseníaspora day 5. But if we focus on Lactobacillus plantarum on its own and associated with SO2, this can control the Acetobacter and Hanseníaspora populations that develop in spontaneous fermentations where no sulfur dioxide nor bioprotective agent was added. From a sensory point of view, or rather the volatilome, it's interesting to see that low concentration of SO2 or the absence of SO2 have characterized the final wines, while we are starting to see that wines where SO2 is used are standing aside, both in association with lactobacillus and on its own. And the dosage at 5 grams per hectoliter is the one that really stands apart. Wines produced with the addition of LPB plantarum and 3 grams per hectoliter of SO2 are differentiated clearly from the rest because of the presence of higher amounts of fatty acids, isoamyl alcohol, methanol, and the total acidity it displays. So there's a contribution brought by these microorganisms. Let me close my presentation because I've run out of time. So actually, plantarum can be an alternative to the use of sulfur dioxide, but I would say that it should be viewed as an important partner with sulfur dioxide in order to attain the goal of quickly eliminating populations of acetic bacteria or unwanted or other bacteria. This hopeless fight on reducing or eliminating SO2 in wines, I think it can be addressed with different approaches by reducing the concentrations of SO2, but also bioprotective agents are added in order to control the unwanted populations. In case you're interested, this paper was published last year in the American Journal of Enology and Viticulture. On top of Lactic Bacillus plantarum, we also worked with a strain of Pichia cluiveri, also supplied by CHR Hansen, with further interesting results. So when you talk about bioprotective agents, you should expand your attention to encompass the microorganisms outside the group of lactic acid bacteria, which are evidently the best known and the most commonly used. Being from Friuli, I'm from Friuli, although I live in Piedmont. Here you see the place where the course of viticulture and enology takes place, I would like to refer to the good wines of Friul. Now the floor goes to Duncan Hamm of CHR Hansen.
Good afternoon, ladies and gentlemen. My name is Duncan Hamm. I'm an enologist originally from New Zealand, but I currently work as an application scientist for CHR Hansen in Denmark. First of all, I'd like to give thanks. Thank you to Emilio and to the organisers for allowing me to have this opportunity to speak today. And a really big thank you to Luca for introducing this topic so beautifully and thoroughly. So I'm going to carry on from Professor Cocolin's talk to talk about how we've brought the first Lactic Plantibacillus plantarum culture for bioprotection in winemaking to market. The use of yeast and bioprotection is a topic in enology that's really come to prominence in the last 10 years or so. It's pretty well known. There's a lot of suppliers talking about it, and there's a lot of products that are open to winemakers to buy. Now, we know that these yeasts can work through passive strategies where they just compete for space, or through active strategies where they actually can produce toxins or actually physically attack other cells. But what about lactic acid bacteria? Do they play a place? I think Professor Cocolin's perfectly introduced that. Certainly in our own company, we produce bioprotective cultures for other industries from lactic acid bacteria. For instance, we produce cultures for the meat industry, which will inhibit listeria. We also produce cultures for the fresh dairy business, which will make fresh dairy products, yogurt and fresh cheeses, double their shelf life. So that caused us to turn our attention to winemaking. Can we investigate and potentially develop bioprotective lactic acid bacteria for wine? And the answer is yes. Where we see yeast and bacteria being quite different is yeast is great for protecting wine during cold soak. Yeast is also really unique because it can help prevent oxidative reactions both through scavenging oxygen and also from the uptake of copper and ions which help catalyze oxidative reactions. In contrast, we see bacteria as potentially having a place in inhibiting unwanted malolactic fermentation, or potentially also, if legislation allows, for protecting grapes and must during shipping. And we see both yeast and bacteria as having potential in preventing high levels of acetic acid, potentially through different mechanisms, but a similar end goal. In terms of the conditions where they're suited, yeast are very acid tolerant compared to lactic acid bacteria. So we see high pH musts is the area where bacteria could readily be applied. And they're also the conditions which tend to lead to bacterial spoilage, whether it's acetic acid bacteria or rogue Enococcus. Both also can potentially play a part in low and no SO2 winemaking. So I'm now going to go on to field trials that we've done with our NOVAT product. But before we go into those, we've actually had this strain in our collection for some time. Its official name is, in the German strain depository, is DSM 27565. And just to highlight the effectiveness on there, I'd like to draw your attention to those two pictures on the right-hand side. In both cases, we have agar plates, which we've incubated, and in both pictures, on the left-hand side, we have a control sample without any protective culture, and on the right-hand sample, we have a sample that's been treated and then plated. I think on the left, it's a wine sample. On the right, it's actually grapes. But it's very, very clear from those. You can see huge amounts of outgrowth of yeast and molds on the left-hand side. On the right-hand side, there is a little bit there, but it's really been controlled by the presence of the Lactobacillus plantarum. And that's interesting, but what about the issues that we see in low sulfite musts, such as acetic acid bacteria or rogue Enococcus? So to help investigate this, we started to do some challenge tests at laboratory scale. Now, this picture is where we've tried to investigate the effectiveness of the plantarum strain against rogue Unococcus. What we did is we took some air juice with 4 grams per litre of malic acid. We ran this experiment in triplicate, and we dosed all of the samples with one of 2 different strains of Unococcus ine. And we inoculated these at 100,000 cells per mil. So quite a high loading, but not a high enough loading for malolactic fermentation to start. For the test samples, we also inoculated with the DSM 27565 strain at 1 million cells. And we can see on this picture we have the malic acid concentration at 3 days and at 14 days, and it's absolutely clear that in the samples, the 2 bars on the left-hand side in each section are the test samples with just Unococcus, and the malic acid has dropped significantly. What's really interesting, on the samples on the right-hand side, where we've dosed with the plantarum as well, for 14 days we've actually been able to inhibit malolactic fermentation. So that's really good. That's at laboratory scale. Likewise, we've also done similar tests to investigate effectiveness against acetic acid bacteria. This example is where we took a grape juice media, added 1% ethanol to it, and inoculated it with a strain of Acetobacter at 1 million cells per mil. On the test samples, we inoculated different strains of Lactobacillus plantarum. I've just highlighted 2 of them here. But the net result after 3 days of incubation at 25 degrees C is a 4 log decrease in the sample with the plantarum, with the Nova plantarum, compared to where the control sample actually ends up in terms of growth. So very, very nice inhibition of acetic acid bacteria. This is all great, but it's at laboratory scale. So we wanted to take it to a commercial scale. And to do this, we ran some trials in Australia. Our first year running these was in 2021, and they worked extremely well. So we actually repeated in 2022 in Shiraz. In the first year, we ran it in Cabernet Sauvignon. Being in Australia, there's not a huge appetite for low sulphur winemaking, so we decided to use a moderate amount, 25 parts per million, total SO2. We had 200 kilograms per tank. We ran each treatment in duplicate, and the wines were either inoculated with the Nova Protect or tartaric acid, or a combination of those. The picture on the right-hand side is the schematic overview. So there's 4 different treatments. The first 2 have tartaric acid with and then without the Nova Protect. And the 3rd and 4th columns have no acetic acid, no tartaric acid addition, again, without or with the Nova Protect. The vinification is very normal, just to help illustrate where we inoculated the different things. On day 0 at crush, we crushed the fruit, added the sulphur, and that's when we added the Lactobacillus plantarum and or the tartaric acid. We then incubated the must at ambient conditions for 24 hours before we added yeast and let the fermentation progress, which from the graph you can see all looks very steady. With these trials, we were able to do PCR, which is a really powerful tool. And first of all, in this picture, this is PCR using a specific probe for this exact strain, the DSM 27565. And the great thing is we actually can demonstrate that we've implanted around 1 million cells per mil of the Nova Protect strain. And what's good, in the 2 treatments without Nova Protect, we basically have no DNA present. So that's great. We know that what we've added has survived and thrived. But what's really interesting is if we look at Unococcus levels. In this graph, the 2 treatments on the left are at low pH, so they have the tartaric acid addition, and we see very little growth of Unococcus in those wines over time. That's kind of to be expected. We know that tartaric acid is actually quite a well-used and well-studied and effective biocontrol. but we also wanted to show that there's also effective bioprotection from the plantarum strain. So if you actually look at the set on the right-hand side, we see even less outgrowth in the one which is at the high pH with the NOVAT strain present, compared to the one that's the third one along, which has no bioprotection at all. It has no tartaric acid, and we see a lot of outgrowth of Unococcus. So it's really clear to see that biostatic effect of the plantarum. The wines themselves look great. We actually put them through a sensory panel in France. And just to summarise, we found this was Cabernet Sauvignon from a cool climate. It does lend itself to green flavours. We found greater fruit weight and less green flavours in the treatment where we dosed NovaProtect without tartaric acid compared to having tartaric acid in the control wine. And we had less bitterness, better persistence and better overall balance through the sensory panel in the one that had NovaProtect. So not only did it protect the wine, but actually delivered a better quality wine at the end of it. So that's really interesting. We're very pleased with that trial set. But again, it was at pretty small scale, 200 litres, 200 kilograms per tank, times 8. This next example we've got is actually a real life example. And when we were launching our NovaProtect product, my colleague Marco Risalti, who's joined us here on stage, managed to have the opportunity to run a trial in a winery where they typically see this picture that we can see on the screen. Just to talk you through it, this is looking at the bacteria levels in a sample after crushing. And the green line indicates the Unococcus concentration. And as you can see, at day 3 and to day 4, there's very rapid outgrowth of the Unococcus. The upshot of that is, represented on this graph, the light green line is the malic acid concentration. And you can see that the malic acid suddenly starts dropping halfway through primary fermentation. By the time primary fermentation is finished, malic has gone through an unwanted fashion, and it's left the wine with a relatively high acetic acid concentration of 0.62. That was the control. In the test tank of equivalent juice or equivalent must, we see a really interesting picture. We still see growth, there's still the presence of the Unococcus, but the presence of the plantarum has restricted its outgrowth. So it only reaches around 10 to the power of 5 CFU per gram, so a lot lower, and it's not enough for malolactic fermentation to kick in. And we can see that on the bottom right-hand graph. The malic acid sees a slight drop, but that's purely from the yeast, because we can see we also have the lactic acid also being measured, and there's actually no increase in that. So the NovaProtect has been extremely effective in inhibiting an unwanted malolactic fermentation in this case. The other good thing about this is the acetic acid comes out lower at 0.55, and if you actually look at the sugar metabolism over time, the curve on the right is the NovaProtect tank. You can see it's a much more even fermentation. It's less attenuated. It finishes a day earlier. And the comments from the winemaker is that the wine is clearly cleaner than the rest. When tasted, it is less oxidative, cleaner than the other tanks. The fruity note stands out, and the tank is among the best in the cellar. Great job, and I would say useful for everyone. That's an awesome endorsement. The last part I'd like to present is an extended, what we would call an extended application of this particular product. We're extremely confident in it. We'd launched the product, but we really wanted to get some data from a winery that had used it across a broad section of their winemaking program. So we approached one of our customers in the Languedoc, in the Corbière region, in the south of France, called CHR Hansen, and they were very interested to see what the product could do in their winery. It's a winery that has a large program of producing preservative-free wine, which then gets traded onto different negotiants. And we took out 8 tanks from the winery. We've represented them here on the screen. and seven of the 8 were treated with the NOVAT Protect, and then we left one as a control, and that was tank number 707, the Musalan. The winemaking was very standard. We crushed the grapes. There was no SO2 added, but at crushing we added the NOVAT Protect, and then the yeast was added just after that as well. With this work, we were also able to apply qPCR. So this is the using the probe specifically for the DSM 27565 strain and nothing else. We're able to determine the concentration early on, and the very good thing that we see, the comfort that we see, is that we actually have very, very good implantation by using this technique. We can see that we're around 1 million cells at inoculation on all of the tanks that had the NOVAT Protect. The other tank here, tank 707, without, shows us trace levels. That could be a little bit of cross-contamination from sampling, but the good thing is it's a trace amount that's significantly lower. So it's very, very clear where the bacteria has gone in and where it hasn't. That's good to know, but what about the protection? Well, when we use PCR and look at the acetic acid bacteria genus, so it's a much broader probe, in every single tank where we added NOVAT Protect, we saw a decrease in the numbers of acetic acid bacteria in the first 24 hours, which is really, really positive. Conversely, in the one tank without the NOVAT Protect, we actually saw a 1 log increase in the level of acetic acid bacteria. But how does that translate into the final chemistry of the wine? Well, this is the the acetic acid development of the 8 different tanks. And as you can see on the top right-hand corner, tank 707, not only does it have the highest acetic acid concentration, but it also develops that acetic acid much more rapidly compared to the others, where the acetic acid development is much more suppressed. So to conclude from this particular piece of work, we saw very, very clear implantation of the Lactobacillus plantarum. Acetic acid development happened fastest and to the highest level in the one tank where we didn't add NOVAT Protect. This can then be linked to the fact that there's a decrease in acetic acid bacteria in the tanks that did have the NOVAT Protect. We didn't show this data because we can't necessarily show everything, but we actually saw no Unococcus outgrowth in any of the NOVAT Protect tanks. That's maybe not initially significant, but it is a cellar that does experience that problem from time to time. So we certainly prevented it from occurring in case it had the opportunity to. And then lastly, again, it's not our data, it's the consultant's data, but all of the tanks that were dosed with the NOVAT Protect graded extremely highly from the winery, so the quality was there as well. And that's obviously what it's all about, is about making good wine. So this is my concluding slide. It's just to conclude and to talk about the product. The product that we've launched from the DSM 27565 strain is called Viniflora NOVAT Protect. It's the first ever bacteria for enology specifically for biological protection. It's a really easy to use product. It's in a freeze-dried format. It's added by direct inoculation. There's no pre-acclimatization or pre-preparation needed. It's a very, very specific dose rate. Now, this is an important point to make. There are other Lactobacillus plantarum cultures commercially available, and we produce some of them, which are for malolactic fermentation. However, the critical number needed for plantarum to drive MLF is over 10 million cells per mil, so 1 times 10 to the 7. However, by being 1 log lower at 1 times 10 to the 6, you can still get very, very effective biological protection. So this product is a lower dose than a plantarum designed for MLF. It also makes it much more cost effective to use and a much more attractive proposition for inoculating the whole winery. So the dose rate is 1 bag per 25 hectolitres or 3.5 tonnes. It's got really nice inhibition against rogue or adventitious Inococcus, which could cause MLF mid-ferment and high VA as a result. It's got good inhibition against acetic acid bacteria. We see in New World countries it could be a very, very strong alternative to using high levels of tartaric acid. And it can be used in no SO2 winemaking, but actually it's also compatible with winemaking with moderate amounts of SO2, say around up to 30 parts per million. So I want to finish off with a few acknowledgements. Melbourne Polytechnic is have the commercial winery where we ran our 200 kilogram scale trials in Australia over 2 years. Enological Vazon certainly helped with this work. Vazon are our distributor in Italy, and their laboratory did a lot of the analysis. In France, Terre d'Expression is the parent company of Cellier Charles Crowe, and I want to acknowledge the fact that they were open for us to share this data, as well as Dubonnet Labs, who we collaborate with to some degree, who are also the consultants involved. I'd like to thank Professor Cocolin and the team at the University of Turin as well. And lastly, I've put Christian Hansen up there because I'd also like to acknowledge colleagues like Marco Rosati for organising a lot of the work in Italy, and our colleague Nicolas Prost, who is our enologist based in France, who also has done a lot of work on this project. So with 33 seconds to go, I'm done. Thanks.
Thank you, Duncan, for your presentation.